Okay, let's try again. So, does anyone know how to identify an Aussie in the audience? Okay. Aussie, Aussie, Aussie! Okay, so they should respond with the oi, oi, oi. That's how you'll know there's Aussies in the audience. I'm an Aussie, and so is Jim. Okay. I see 1, 2, and Aussie, Aussie, Aussie! All right, so now you know. EOS Biosciences, I'm their CFO. We're a bio-targeted nanomedicines company. We're based in Los Angeles. In fact, we've got a worldwide exclusive license from technology developed at Cedars-Sinai Medical Center right here in Los Angeles. So we're kind of born and raised, unlike me, in Los Angeles. We have a product, a technology, which we call eosomes. And these eosomes are a small nanoparticle. And what they do is they entrap and encapsulate a chosen therapeutic, and they deliver it specifically to the targeted disease cell in a targeted way, and then very specifically to the actual disease cell, releasing the therapeutic once it hits the target. It's not really rocket science. It's just genetic engineering is what we're talking about here. In the top part of this slide, what you see here is just a basic representation of a protein. And the eosomes, the foundation of the eosomes is a protein with 3 specific kind of domains. The first domain is this targeting domain. Okay? And through genetic engineering, we've created this protein out of one piece of a protein. We've selected this particular peptide to be on our particular eosome protein. The second piece of the protein is this membrane penetration and intracellular trafficking peptide. And what that does is it helps to release the therapeutic inside the disease cell. And the third peptide, the third piece of this just simple protein, is this therapeutic binding peptide. So what that does, it helps to bind to the therapeutic. When the therapeutic is joined to this protein, it self-assembles into an eosome, which is about 50 nanometers in diameter, very small, and it's able to penetrate tissue, and it's able to get inside the disease cell very quickly, release the therapeutic. And there's a graphical representation of that process. There's the targeting aspect, it goes to the disease cell, it enters into the cell, it releases the therapeutic, and the therapeutic does its job. The point is that it can be very effective, and it's because the therapeutic's being delivered specifically to the cell, and it also enhances the safety profile of the therapeutic. For a lot of cancer therapeutics, they're very toxic,